Today's OCCPR webinar will be about NCI's antibody program. Dr. Tara Hiltke, a program director in OCCPR, will talk about the program, the portal, and our current request for target submission callout. We are glad you can join us.
Thank you for joining the NCI's Office of Cancer Clinical Proteomic Research webinar series. As a part of NCI's Cancer Continuum, OCCPR mission is to improve prevention, early detection, diagnosis, and treatment of cancer by enhancing the understanding of the underlying molecular mechanisms while advancing proteome and proteogenome science and technology development. Through the integration of genomics and transcriptomics with proteomics, also known as comprehensive proteogenomics, OCCPR's aim is to advance basic, translational, and clinical research through OCCPR-supported programs. This webinar series hopes to educate you to all the resources OCCPR and its researchers have to offer.
Thanks, Anna. Hello and welcome to the NCI Antibody Program webinar. My name is Tara Hiltke and I'm a program director within the Office of Cancer Clinical Proteomic Research. Today, during the webinar, you'll be muted to minimize the background noise. However, you can take advantage of the chat feature on the WebEx, or you can email me directly after the webinar for any questions. If time permits, we'll answer as many of the questions that we've already received or from the chat feature as we can. So we'll get started because there's a lot to discuss today. First, I'm going to give a little bit of a background about the program. The NCI Antibody Program within our office was developed back after a Reagents and Resources workshop back in 2005, which emphasized the importance of making well-characterized affinity binders, such as antibodies, and making those available to the community. The NCI Antibody Program was launched in 2008, and initially it used recombinant proteins, which were produced by Argonne National Laboratory to make about 100, to make antibodies to about 100 putative cancer targets. In 2011, however, we changed to a more fit-for-purpose approach and opened up the antibody production to members of the scientific community. This resulted in targets which were produced, screened, and selected in coordination with the end user and their intended application. I'm going to get more into the application process towards the end of this webinar, but first I wanted to walk you through the Antibody Portal and all of its features. So, what you should be able to see currently on the WebEx is our Antibody Portal homepage. You can get to this by typing antibodies.cancer.gov into any browser. As you can also see at the top with my mouse, we also have access to some of our other resources such as the Assay Portal and the Data Portal. We won't be discussing those today as there's not enough time. However, feel free and I encourage you to explore those additional resources. Let's see, I also want to point out on the home page that we have two buttons here which are associated with our antibody program. The first one that I want to talk about is the antibody characterization process, so I'm going to click that button. All antibodies in our program are characterized at the Antibody Characterization Laboratory, which we often refer to as the ACL. The ACL is a laboratory within the Frederick National Laboratory for Cancer Research, and this laboratory is directed by Dr. Simona Colantano. The goal of the NCI antibody program is to produce 3 monoclonal antibodies for each full-length protein antigen and 1 antibody for each peptide antigen. The antibodies are screened and selected first for the applicant's intended end use, which we call fit for purpose, and then they're further characterized at the ACL using methods such as ELISA, Western blot, immunohistochemistry, affinity measurements through SPR and other methods, as well as immunoprecipitation. All the data, both positive and negative, is made available to the scientific community through our web portal. All of our antibodies are also available for research use through the Developmental Studies Hybridoma Bank at the University of Iowa. A subset of our antibodies are also distributed by commercial vendors, and this is done through various licensing agreements. I would now like to move to the Antibody Portal and its navigation by returning to the home page, clicking back, and now choosing the Available Antibodies button. Okay, so this is the default page for the Antibody Portal itself. Most people prefer to browse by antibody, but as you can see here, there is a toggle feature which allows you to also browse by antigen and changes the display slightly. Since most people use the browse by antibody button, I'm going to go back and focus mainly on that. The antigen information is embedded within the antibody area, so there are certain unique aspects within the antigen part, but most people that are interested in the antibodies, this is where you would focus. Okay, so as you can see, we currently have 583 antibodies available, and these are all arranged alphabetically, as you can see here, with up to 24 pages of information. And looking through each one of these could take a while. So our portal has several ways in which you can narrow down your search. There is the ability to filter using these categories on the left, which I'm going to get into in a minute. And that allows you to kind of filter it down, especially if you don't know which specific antibody you're interested in. Maybe you're interested in something that has a specific characterization or a certain size. So that's where you can kind of filter on this part. Additionally, if you know you are interested in a specific antibody, then you can search using this search bar up here. And this will take most common known aliases. So for instance, if I type in HER2, you see it comes up, even though we use the more conventional mechanism for the naming it ERBB2. So that's how you would search. Now let's examine the different ways you can narrow your search using the filtering mechanism. First, I want to show you kind of how you can filter by antigen recognition. So we'll just select peptide as an example. So we're going to go from 583 down to 274 antibodies. These 274 antibodies were produced using peptide antigen targets, which then obviously recognize the peptide. If, for instance, we are more interested in those that had recombinant, we can choose by selecting this, and you get 376. Okay. Be sure you unclick your prior selections, or else it will continue to narrow information based on all the selections that you have. Another way to filter is by characterization type. So that's in the second section. If you're interested in, for example, tissue IHC, you can click on that. And like I've said before, all of our positive and negative data appears on our portal. However, if you're only interested in the options which had positive outcomes, you can just simply deselect the negative button, and it will show you that we have 86 entries that have positive outcome from a total of 180 that were tested. Okay. You can also filter by isotype if that's of interest, or the different types of affinity binders. We use mouse or rabbit typically, but I'm not going to click through those just in the sake of time, but they will do similar features. Another thing I wanted to point out is that under our external links, you can also filter by the CPTAC assay portal. So if I click that, selecting this then narrows to antibodies on our list which have qualified assays. So for example, if I click this first one, you'll see here a link to the assay portal. If you click that, it'll take you over to the assay portal, and you can see, it might take a minute. And then you can see the assay that's associated. So I'm going to go back, and that would be for immuno-MRM assays associated with that particular antibody. So now I'm going to go back to the filter page, and now I'm going to unselect the CPTAC assay portal, and I'm going to now show you our publication feature. So this is something we added very recently within the last few months. And it's still being populated, but I wanted to show you how it kind of works. So if you click on the publication tab, a bunch of PMIDs appear. If you hover over the PMID, it will provide you with a title that's for that particular publication, which kind of lets you kind of look at the different categories so you can decide if these are of interest to you. So for instance, if you're interested in this third one, which is based on papers for DNA damage response, you can just unclick the other ones, and now you scroll up and you have 51 of our antibodies which were featured in this particular publication. Okay, so unclick that now. And then lastly, we also recently added these thematic panels, which is another way to look at the antibodies in kind of like a grouping. And so we currently have antibodies that kind of fall into, we have a bunch of one-off antibodies, but we do have some collections that fall into these thematic panels, specifically the DNA damage response pathway, the epithelial mesenchymal transition, and the RAS pathway. And so, you can click that, and then you can see that we currently have 123 antibodies that are in some way associated with the RAS pathway. All righty. Now I want to explore a little bit the organization and the characterization data for our antibodies. First, I want to point out that you'll notice that each of our antibodies has this CPTC prefix, which denotes our program, but also, more importantly, it is a way to ensure that the antibodies aren't being sold under different names. Anyone who publishes or sells using our antibodies must keep the CPTC name as the clone name. Let's select an example. I'm going to choose this one, AKR1B1-1, as it's a good example of a well-characterized antibody. All right, so if I click on that, I just want to point out that you're also going to see the two sister antibodies listed. So you're going to see the -1, -2, and -3 here. That's because they were each used a common antigen to create the antibody, so they're all kind of clustered together. Each antibody will have background information, as you can see here, which includes our catalog number, what we call it, what the original target antigen name was, what its isotype, what species it was made in, as well as like what the immunogen was. In this case, it was a recombinant full-length protein. Okay, so then I next want to show you, and one other thing, just like before with the assay portal link, if there is an assay portal or a publication, that would also show up here with a link. Okay, so now by clicking the plus sign of each method, it will open a window of the different characterization data associated with that particular antibody. So if I open up the cross-reactivity data, I can see that. Additionally, for IHC, I see a tissue image, and also the result will either say positive or negative. So that's another way to see. In some cases, it might show something, but you'll see how we designate it as positive or negative. So for this particular antibody, we have a variety of characterizations that span from ELISA to Western blot to IHC. And then similar data is also seen for the -2 and -3 antibodies, which allows you to kind of compare the different ones from the same immunogen. And at the bottom of the page, we have information on the antigen. I'm going to scroll down here, which includes the various, in addition to the name and our CPTC name, it also includes some keywords and some general functional information, what system it was produced in, if that's available. It also includes the sequence, the molecular weight, and SDS page gel denoting how it looked when we received it. Okay, so I also wanted to mention that if you're looking at an antibody and you don't see all the data that you're expecting, please email us and ask, as they may be in queue for upload or still in characterization. We try to enter the antibodies as soon as we have the initial characterization completed, and then we continue to populate the portal as the additional data becomes available. Some of the characterizations that we do at the ACL can be run in parallel, while others, such as endogenous testing, require prior information to proceed and thus take more time for us to complete and add to the portal. All right, so after reviewing the data, if you're interested in purchasing the antibody, you can scroll back up to the background information, and there should be a link for how to purchase. So if you click this, it'll take you to the Developmental Studies Hybridoma Bank, which sells all our antibodies. This intermediate page will pop up if you click the Purchase Now button. It'll take you, it tells you you're going outside of the website. It takes you to DSHB. And the link takes you to the specific antibody at DSHB that you're interested in. This is important because DSHB sells 50,000 antibodies, so this now takes you to the one from our portal that you're interested in. If you're interested additionally in, while you're on DSHB's website, if you're interested in learning more about our CPTC antibodies, if you click their home button, we're actually featured as one of their collections, so we're the NIH CPTC. So if you click that, it brings you to kind of our overall collection that they are selling. Okay, you may notice that the DSHB doesn't have all of our antibodies listed, as several of them are in queue or require production requests. There's always a little lag as we try to coordinate our characterization with their production, the QC and the uploads to their site. Okay, so I'm going to go back to our portal now. Alrighty. The other thing I wanted to point out is on the main antibody browse page, you also see this purchase feature with the shopping cart. So if you just click the shopping cart, it's going to take you to the same intermediate page. So there's two different kind of ways to find how to purchase the antibody. All right. Next, I want to spend a few minutes to talk about the NCI target request application process. Approximately once a year, we have a call out to the scientific community for cancer-related targets to make antibodies. Currently, we have an open call which ends on February 13th. And this is a resource provided by the NCI which pays for the antibody production and characterization within the ACL. And if the applicant is selected, they only need to provide the immunogen, antigen, and any counter screen proteins or peptides that may be needed. So how do you find that on ours? If you go up here to where it says Antibody Portal Home, now you're back to our main home page for the portal. Over here on the right, there's a thing that says how to apply. So if you click that, it brings you to the background information. This large paragraph gives you kind of a lot of what I've already discussed, but additional information about the NCI antibody program. But if you scroll down to the bottom, you can see here that there's a link for the online application, as well as my contact information. So if you have any questions, you can send me an email or give me a phone call, and I'll be happy to help you. So let's just look at what the application entails. So if you click here, you'll see that it's a very short application. Shouldn't take you too long to fill out. Just simple information, who you are, where you're from, contact information. We also ask for the investigators to submit information on funding. We want to ensure that the people who are receiving the antibodies through our program are actually going to be able to use them because they have funded research. We also want you to give us information. Do not provide anything proprietary. So here you're going to describe the antibody, why it's important. Then if you've ever used any of the commercial antibodies that are available, please tell us your experience. Maybe you've tested the 10 that are available, they don't work, thus you need a fit-for-purpose antibody for your application. Then you're going to describe what your intended use is. That's here for Western blot, IHC, etc. We want to have information on the antigen. This allows us to determine, at least initially, how we think it's going to be produced, whether it be in mouse or rabbit. We also want to know, whether it's in E. coli, mammalian, is there post-translational modifications? So all these questions are here and available. If you have additional characterizations that you want to show us about your antigen, you can email them to us because obviously you can't upload them on this form. And if you have any publications associated with it, that's sometimes helpful for the review. Then you're going to just check off that you agree to the following statements, and then you'll submit your request. Okay, so let's go back. So let's say so as you can see, this application is pretty straightforward. It's not too long or cumbersome. The applications are all reviewed by an external scientific committee who verify first that the targets would be useful for cancer research. They review the number of current antibodies commercially available to the same target to determine if additional antibodies are needed. They review the answers that you provide to help them to prioritize the projects for the NCI to pursue. If an applicant's project is chosen, then an MTA is required. Oops, I'm sorry, I've got to go back into the application. The MTA is actually down here. So if you wanted to review the MTA with your tech transfer group before you decide to apply, you can just click it and it's available to you. In a very high level summary, I'm going to just say kind of what the MTA states, which is that all the antibodies have to be deposited at DSHB for research use, and it denotes that the DSHB only sells the antibody and not the hybridoma or the clone, and that one vial of the hybridoma or clone is provided back to the applicant for internal research or commercial purposes. It also states that all publications must use the program's designated CPTC name. And again, to remind you that this is for us to track the antibody not only through literature, but it allows the end user to know that the antibody is, even if it's being distributed by various sources, it's the same antibody. So you're not buying the same antibody with different names, only to find out later that they're the same. Once this MTA is in place, the next step is to have a conference call between the scientific participant, the NCI, me, as well as the ACL laboratory. And this would be held to discuss the project and all specific requirements. This is where we discuss the intended use of the antibody, how to best screen and select for it. This understanding is critical for developing appropriate immunization, screening, and selection strategies, and improves the likelihood of creating an appropriate fit-for-purpose antibody that you will want to use. The participant also then sends the recombinant protein peptide to the ACL laboratory, where they'll do some quality control to make sure that it is doesn't have too much endotoxin, that it is it's a clean antibody. We can't have things that have too much background proteins. That limits the success. Again, we encourage the scientists to participate in all stages of this process, specifically in the antibody screenings, counter screens, and selections. The goal is a fit-for-purpose antibody for the participant and ultimately the scientific community. Additionally, the ACL will characterize the antibodies using its characterization pipeline that I talked about in the beginning of this webinar. This will provide a broader understanding of the antibody use and what things it works or doesn't work according to our SOPs. For example, if we receive a peptide target for an immuno-MRM application, that's a very limited use. However, we have found that by running this same antibody through our pipeline, we often identify antibodies that will also work for Western blot or immunohistochemistry, and this gives more utility to the antibody. It doesn't remove the fact that it is a fit-for-purpose antibody for the applicant, but it makes it broader to the scientific community. Okay, as discussed, all the data, positive and negative, goes on our portal. This doesn't mean that that things that we say are negative couldn't be made positive by tweaking protocols and things, but it is saying that the protocols that we routinely use for our characterization pipeline have demonstrated it to be positive or negative, and we do provide those SOPs on our portal so you can review how we did it and decide whether or not you want to try a different blocking agent, etc. But it allows you as the user, when you're trying to decide which antibody to purchase or to examine further, that you can kind of narrow down your search using some of our data as like preliminary information. Okay, so now I want to switch. I know we don't have a lot of time left, but I do want to answer a few questions we've received. So if you have any additional emails or any additional questions that we don't get to, feel free to email me anytime. Happy to help with that. Okay, so one of the questions that we received was, can I apply for more than one target antibody? The answer is yes. You can apply for as many as you want. Please, however, prioritize your selections in your application so the committee will be able to kind of know what you're thinking is which ones are top on your list for, to pursue, because the resources may not allow for all of the ones that you're submitting to be selected. Okay, another question was, can I purchase these antibodies for non-cancer research? Of course. We feel that the targets often bridge between many diseases and research interests, and we encourage you to examine as many of the different antibodies that you see that seems to fit into your research plan. Okay, another question was, how can I get these antibodies for commercial use? Okay, so this isn't a super straightforward answer, so I'm just going to say that most of our antibodies are available for commercial use. This is through a licensing or other mechanisms that we have in place. It's too much to kind of get into today, but if you are a commercial, if you're on this webinar and you're from a commercial institute or commercial place and you want to be able to also distribute our antibodies, just contact me directly. We can set up a conference call and I can kind of walk you through kind of the different ways with which you can obtain the materials. I think that's all the questions I had as, let me see if there's any in the, I guess that's it. So if, let me see, I'll see. Okay, so somebody asked about whether or not we could do flow cytometry as an application, and of course we can. We don't necessarily do that routinely in our characterization laboratory, but this is why working together with the scientist who submits the application and wants to test it for their end use applications, this is a perfect example of how that would work. We would work with you to utilize your flow cytometry expertise, and then we would add value through our other characterization methods that we have. Let's see. Trying to see the questions kind of, I don't know, trying to pull up the questions on the WebEx, but I'm having a little trouble. So I think that might be the only one. But like I said before, if you have any questions, please reach out to me. My email is here. It's also on the application. And I'm happy to talk with you about any of your needs, questions, how to help you. Thank you.
Thank you for joining the Office of Cancer Clinical Proteomics Research webinar series. Please visit our online resources included here and take a minute to answer our online survey. The Office of Cancer Clinical Proteomic Research is part of the U.S. Department of Health and Human Services at the National Institutes of Health, National Cancer Institute. For more information about OCCPR, please visit www.proteomics.cancer.gov. For general information about cancer or clinical trials, please call 1-800-4-CANCER.